Welcome to Roadmap to Residency Evidence-Based Applications. Hello, everyone. I'm Abhiras, and in this video, we will be discussing some important things about enzyme structure and functions and how the functions of the enzymes are affected by different situations. So the common features for enzymes and organic catalysts are they catalyze only thermodynamically possible reactions. They are not used or charged during the reaction. They don't change the position of equilibrium and direction of the reaction, and they usually act by forming a transient complex with the reactant, thus stabilizing the transition state. The specific features of enzymes include they accelerate the reactions in much higher degree than inorganic catalysts. There is specificity of action. They have sensitivity to temperature and they have sensitivity to pH. So the structure of enzymes. So enzymes, they are simple, which contain only protein, or they can be complex or hollow enzymes, which contain protein part and non-protein part, which is also called as a cofactor. So the apoenzyme, which is also called as a protein part, and there is a cofactor. Now the cofactor may be a prosthetic group or maybe a coenzyme. So prosthetic group is usually a small inorganic molecule or atom, and they usually tightly bound to the apoenzyme. The coenzyme is a large organic molecule and they are loosely bound to the apoenzyme. Okay, so apoenzyme, which is a protein part. So the coenzymes act as a group transfer reagents, like so hydrogen electrons or groups of atoms can be transferred. The coenzyme classification, they can be metabolite coenzymes or they can be vitamin-derived coenzymes. So vitamins cannot be synthesized by mammals but must be obtained as nutrients. That's why because of the coenzymes. So the vitamin-derived coenzymes, the vitamins are required for coenzyme synthesis and must be obtained from nutrients. So the most vitamins must be enzymatically transformed to the coenzyme. So the deficit of vitamin and as a result, correspondent coenzyme results in the disease. So the enzyme's active site, so the substrate usually is relatively a small molecule and the enzyme is a large protein molecule. Therefore, the substrate binds to a specific area on the enzyme only. The active site is the specific region in the enzyme to which substrate molecule is bound. So the properties of enzymes or specificity of enzymes. So there's absolute, relative, and stereospecificity. So absolute specificity is in one enzyme acts only on one substrate. For example, the urease decomposes only urea, the arginase splits only arginine. So that is called as absolute specificity of enzyme. The relative specificity of enzyme is that when one enzyme acts on the different substrates which have the same bond type. Example, the pepsin splits different proteins. Whereas stereospecificity means that some enzymes can catalyze the transformation only substrates which are in certain geometrical configuration, example, cis or trans, then that is called as stereospecificity. Sensitivity to pH. Each enzyme has maximum activity at a particular pH, which is called as the optimum pH for that enzyme. So for most enzymes, the optimum pH is near 7, and there are some exceptions. Here we can see the activity of trypsin and the activity of pepsin. The activity of trypsin, the most highest activity is at the pH nearly at 6, okay? And for whereas for pepsin, the activity of pepsin decreases as the pH increases. So sensitivity to temperature. So each enzyme has maximum activity at a particular temperature, which is called as optimum temperature for that enzyme. So enzyme will denature above 45 to 50 degrees Celsius. Okay. So the most enzymes have a temperature optimum of 47 degrees Celsius. Here in this picture, we can see that at 47 degrees Celsius, there is optimum enzyme activity, and at 45 degrees Celsius and beyond, there is denaturation of the enzymes. So there are 6 classes of enzymes. So number one class is the oxidoreductases. They catalyze the oxidation reduction reaction. You know that oxidation is loss of electrons and reduction is the gain of electrons. So they are oxidases, peroxidases, and dehydrogenases. So the transferases are the enzymes that catalyze the group transfer reactions like alanine transaminase. So hydrolysis are the enzymes that catalyze the hydrolysis reactions where water is the acceptor of the transfer group. For example, esterases, peptidases, and glycosidases. Lysases catalyze the lysis of a substrate and generating a double bond in a non-hydrolytic or non-oxidative elimination, like pyruvate decarboxylase. So isomerases are the enzymes that catalyze the isomerization of molecules, like alanine racemase. And ligases are the enzymes that catalyze the ligation or joining of two substrates, and they require the chemical energy like glutamate synthesis and all. So the isoenzymes or isozymes, so some metabolic processes are regulated by enzymes that exist in different molecular forms and that are called as isoenzymes. So isoenzymes, so multiple forms of enzyme which differ in amino acid sequence but catalyze the same reaction. They are called as isoenzymes, but they can differ in kinetics, they can differ in regulatory properties, and the form of coenzyme they prefer, and the distribution in cells and tissues. And these isoenzymes are coded by different genes than the enzymes. So we have here a 60-year-old patient presents to the clinic with symptoms of indigestion and discomfort after meals. Upon further evaluation, the physician suspects a gastric ulcer and decides to conduct an investigation into the role of gastric acid in ulcer formation. The patient's symptoms worsen when consuming acidic foods. Which of the following best describes the effect of pH on enzyme function relevant to this patient's condition? Enzyme activity increases as pH approaches neutrality. Enzyme activity is highest at extreme pH values. Enzyme activity is optimal at acidic pH values. Enzyme activity is optimal at alkaline pH values, or enzyme activity is not affected by pH. So enzyme activity is optimal at acidic pH values. So in the context of gastric ulcer formation, the enzyme relevant to consider is pepsin, which is secreted by the gastric chief cells in the stomach. So pepsin plays a crucial role in the digestion of proteins, particularly in the stomach, where the acidic environment facilitates this activity. Pepsin is most active at acidic pH values, typically around pH 2. And this acidic environment in the stomach is necessary for pepsin activation and optimal enzyme function. In conditions of high acidity, such as after consuming acidic foods, pepsin activity increases, which may exacerbate the symptoms in patients with gastric ulcers. Thank you for watching. Keep studying.